happen through mutation, it can happen through anything that can change the Hardy-Weinberg equilibrium. Is it clear? Am I correct, Mamuna? Do you agree?
Yes.
Yeah. Okay. Continue. Yes.
Then it's speciation, in which new species are found from which are evolved from pre-existing species with
Yes.
new characteristics found in them.
Yes. New characteristics and new or different phenotypes. Okay. Then we discussed about proof of speciation. Okay. For which let Mamuna take over. Mamuna, what was the proof of speciation experiment?
Sir, I was absent the last class.
Okay. Okay. Abia, you were there?
No, sir, I was not there in the last class.
Mariam, who was there in the last class? Mariam and Nabil have joined. Okay, welcome. Good evening, Mariam and Nabil.
Good evening, sir.
Yes, Mariam. Were you there in this class?
No, sir. Last class, I guess towards the end, I got disconnected.
But this is not from the last class actually towards the end. This is proof of speciation. Remember the microbial experiment that pre-existing advantageous mutations, they are carried forward, which gives rise to new phenotypes. And
No, sir. I wasn't present during this part.
okay, so Saifullah, I think then you only have to continue as you were doing. Yes, tell us. Am I audible to you, Saifullah?
Yes, yes.
Yeah. Continue.
Like, the proof of speciation is shown like pre-existing mutations, species that have mutated due to environmental changes and climatic changes, and the new species that we have present today. By comparing the bone structures and skeletons, we can say that these species which are present today have been like this in the past, millions of years ago.
Right. So that's about what we are observing. But you remember the proof was more subtle. In this experiment done in microbes, we found that microbes can evolve faster, right? Because they have very short lifespan and life cycle. So any mutation can just be carried on to the next generation very fast, right? Or any change in the allele frequency or any change in their genome. And over time, if these advantageous changes keeps on accumulating, they can evolve faster. So within a few years, we can see, or within a few months, we can see new species developing in microbial experiments, right? In bacteria. You understand?
Yes, sir.
Yeah, and then we went on to do natural selection. What was natural selection?
Sir, I think we did this before also, sir. Like last class.
Yeah, I think you explained it, right?
Yes, sir.
Yeah, that's why, because many students were absent, so that's why we are just going through it quickly. So Nabil, would you like to take natural selection and then the summary for plant and animal?
Sir, can I do it for humans, sir? I made like a presentation. I collected all the pictures.
Okay, so you can do it for humans in the end as well.
Okay, sir.
Because others were not present in the class, so there are not many people who can speak for this chapter. So you can give a quick summary for them. It will help them as well, and we'll finish this chapter.
We did an in-class assignment about summarizing the pictorial evolutionary graph. So based on the graph, we summarized the evolutionary lineages of angiosperms, conifers, ferns, and bryophytes. So from this, we found out that angiosperms have the most latest characteristics, like the most newest ones, whereas bryophytes have the most primitive characteristics. This is because they have undergone very less branching descent over the past, very less branching descent. And ferns, conifers, they are the oldest, one of the oldest species that are still existing. to this date.
Yes.
And the bryophytes came up during the Cenozoic. The ferns and conifers and cycads, they came up during the Mesozoic. And moreover, these bryophytes came directly from the chlorophyte ancestors, whereas these ferns, conifers,
Chlorophyte means the ones which were the first primitive plant ancestors that were capable of doing photosynthesis. Yes.
And these ferns, conifers, and cycads came up during the Mesozoic, whereas the angiosperms came up during the Cenozoic. And most of the plant species that we see these days, they came up from xylophyton groups.
Yeah,
Then,
the latest one.
yes, sir.
Yes. Okay.
And then we did
Animals.
brief account of evolution of animals. The first cellular forms of life evolved around 3.2 billion years ago. The non-cellular aggregates of molecules or inorganic molecules are assembled into cells within membranous coverings or envelopes. Some of the early unicellular organisms evolved the ability to release oxygen and start making the environment less reducing. These organisms which could release oxygen had evolved chemical reaction pathways similar to photosynthesis. Then we did an in-class assignment about the evolutionary lineages of animals.
So the
We did
point that you just said, Nabil, about the photosynthesis, that's more for apt for the plant ancestors, right?
Yes,
Though they
sir.
were organisms, but yeah. Yes, continue.
Then we did for an in-class assignment for animals. So from that we found out that the turtles are reptiles. They have evolved from the sauropsids, which have evolved from the early reptiles. They possess the most primitive characteristics. Then lizards, they are also reptiles. They have evolved from the sauropsids. They have had the most branching descents. And the crocodiles, they're also reptiles. They have evolved from the thecodons. Their closest ancestors, for them, the closest ancestors are dinosaurs. Their main branch is the sauropsids. And the birds, they are the aves. They have evolved from the thecodons. They are the closest ancestors to the dinosaurs. And their main branch is the sauropsids. And is the mammals. Mammals, they have evolved from the therapsids. Their main branch is the synapsids, and their closest ancestors are the birds. And we also saw why
No,
amphibians
for mammals, we can say that like evolutionarily the closest ancestors are not birds, because birds are closer to reptiles. Mammals, you see, there is a complete different branch of synapsids, because mammals are viviparous. We give birth to the babies, but birds, reptiles, amphibians, and fish all lay eggs, right?
yes, sir.
So here it's the parallel. It's, if you have to go for birds, do you see any branching here which is connecting it to birds? You have to go all the way back to synapsids and then sauropsid branch, and then you have to go to this thecodont, and there you will see dinosaurs, and from dinosaurs emerged birds. So they are just evolving with us simultaneously on the planet. We are also here, birds are also here, dinosaurs are gone, but it does not mean they are our closest ancestors. Okay.
Yes, sir.
Yes. Okay.
And then we saw the why amphibians and fishes have not present in the graph. This is because they started way before the first reptiles. The closest living organisms to dinosaurs are the birds, and we also saw the closest ancestors to mammals are therapsids. Then the first invertebrates were evolved around 500 million years ago. The first jawless fish evolved around 350 million years ago. Proper fish with snout and fins evolved evolved that were dependent on both land and water, evolved into the amphibians. Then these amphibians evolved
Yes.
into reptiles in around 300 to 350 million years ago. Then we saw the major differences between amphibians and reptiles. These amphibians, their life entirely depends on water, whereas reptiles, their life is practically independent on water. They have thin-shelled eggs.
No, amphibians' life do not entirely depends on water. In fact, an amphibians' life depends both on land and water. They lay eggs in the water, but they live and they breed on the soil, right?
Yes, sir.
That's why they give these very fragile shells, very fragile shell-less eggs. The eggs of frog we were talking about in the last class, remember?
Yes, sir. So amphibians, their lives partially depends on water.
Yeah.
Reptiles, their life is independent of water.
Yeah.
The amphibians, they lay thin-shelled eggs, and reptiles, they thick-shelled eggs, which can survive the heat, the sun. Then
Okay.
land reptiles became dependent on land. The land reptiles were then known as dinosaurs. These reptiles then evolved into birds. Some of the proofs are Archaeopteryx, a fossil link between reptiles and birds, that is dinosaurs and birds. And the first mammals came from the synapsids. The first mammals were like shrews. They were viviparous and were more intelligent in sensing the environment and adjusting according to the change. So
Yeah.
reptiles on the other hand were cold-blooded, whereas mammals were warm-blooded organisms. So when reptiles and dinosaurs became extinct, mammals took over. Some mammals have adapted to water. Then we looked at the
Yes.
origin and evolution of humans.
Yes. Not only mammals have adapted to water, many reptiles and many birds have also adapted back to water. Right? So they came out of water. Their ancestors started living on land, but some of them went back to water to get adaptation. And that movement back to water happened because of many factors, and scientists are studying that. For example, competition on land, more options in water, things like that. So you must have seen this behavior of many birds who cannot find food on land. Then what they do is they hunt fish from the water. They actually dive in the water, go very down, take a fish in the beak, and then come up. Have you seen this?
It's a
Have you seen?
seagulls.
Yes. So they are, as birds, they are also adapting to water. You know, they are living more and more closer to water. They are hunting in water. They are getting food in water, and they are also laying eggs around water. So that is what started happening for these reptiles, birds, and mammals as well, who now live in water. Okay, aquatic reptiles, aquatic birds, and aquatic mammals. Okay, yes. Now let's come to the last part. Yes, Nabil.
Then we looked at the origin and evolution of Homo sapiens. So about 15 million years ago, the primates named as Dryopithecus and Ramapithecus emerged. So then we look at their characteristics. So first, Dryopithecus and Ramapithecus, their body was covered with hair. They walk like chimpanzees do. It is believed that they live together. So
Yeah,
Dryopithecus
they
was
walked even lower than chimpanzees. They walked more like bears. How bears walk. They were not at all bipedal. They were walking on all their 4 feet. Okay.
so
Chimpanzees
Dryopithecus.
sometimes can also walk on 2 feet. If you see them in their own habitat, they can manage to do that. They don't do it often. They have a curved body, but they can do that, right?
Yes, sir. So Dryopithecus was more ape-like, whereas Ramapithecus was more man-like. Then, so then we look at some hominid characters. So the first fossils that showed hominid characters were discovered from Africa, from Africa, that is Ethiopia. So the first human-like primates evolved around 3 to 4 million years ago. They were called as the Australopithecus, that is about 2 million years ago. So they were about 4 feet in height. Fossil records of Australopithecus have been found from the East African grasslands. And
Yes.
then we looked at the first hominid species, was called as the Homo habilis. Then it came about 1.8 million years ago, and 1.8 million years ago, it was fruit eating. It ate tubers and roots, so it had bigger brains and than earlier, but it was still smaller compared to the present humans. Then we looked at the Homo erectus. The Homo erectus came up about 1.5 million years ago. Okay, so Homo erectus came up about 1.5 million years ago. So they had a larger brain, about 900 cc, and they were hunters. Then we looked at the Homo neanderthals. So the Homo neanderthals came up in Asia. They had a bigger brain, about 1400 cc, and they came up about 1 lakh to 40,000 years ago. They were also called as Cro-Magnon man. Then after that, finally came the Homo sapiens. The Homo sapiens came about 75,000 to 10,000 years ago. Proofs of human settlement in agriculture were found around 10,000 years ago.
Yeah, right. So we have evidences as back in time as 75,000 years ago for Homo sapiens, but the evidences for agriculture and settlement goes, around the same time. So we know that Homo sapiens have been around, and they were overlapping with Neanderthals, right? And Neanderthals were also overlapping with few other Devisonian and other species about which NCERT does not talks about, so it's not in your syllabus. Okay. Apart from sapiens and Neanderthals, after Homo erectus, there are many more species. Not many, but few, a handful of species that we are figuring out. Okay. So by this chapter is clear, everyone. Anyone has any doubts with respect to anything in this chapter?
No, sir.
Okay, very well. So there's one thing that I want to announce before we move forward. So tomorrow you will have your test. It's a mandatory 1 hour long test for your boards and school exams based on chapter 6. So please note down this. Chapter 6 is before evolution, because we just finished the last class of before chapter. What was the name? Molecular basis of inheritance, right? Please note this down, and all of you have to attend and give the test, and no excuses that you were not present in. This has been done before. So chapter 6, it will be a 1 hour test, 25 marks subjective, and there will be total 10 questions. Okay, 5 will be 2 markers and 5 will be 3 markers. Okay. So total 10 questions, and you have to finish it and compile it as a PDF and send it in the group to me. Okay. So everyone, tomorrow in class. Is it
Yes, sir.
clear? Mariam, Saifullah, Wafi, everyone has noted down this. Mamuna, Abia, Madiha.
Yes, sir.
Yes, chapter 6. So revise it completely, a full chapter. Total, overall. From the beginning till DNA fingerprinting. Okay, very well. So let me start with the new. So we're going to start with chapter 8 today. I think, okay. So everyone again tell me which chapter is coming in everyone's syllabus after chapter 7. Is it 8? Human health and disease? Should we go for that first?
For me, it is on term 2.
Sorry? For you, it's in term 2, you mean to say.
Yes, sir.
So what's
Sir,
coming
for
in
me,
term
this
1?
chapter is completed.
Chapter 8.
Yes,
But it's coming,
completed,
right?
yeah.
Yeah, so this is the next chapter that we should go. Anyone who has like some erratic chapter 10 or 12 coming in, but 8 is not there? Anyone?
No, sir.
Okay, so now is the time.
Sir, in
Yes.
my school, they started with the 9th lesson.
9th chapter?
Yes, sir.
They have done chapter 8 or they have omitted chapter 8?
Our ma'am told like she will take it after the.
After 9th. Okay, anyways, it will be there, right, in your exam.
Yes, sir.
Yeah, so we'll do chapter 8 then first, because it's important to do chapter 8 like the way I have planned the syllabus. If chapter 8 is not done, then chapter 9 becomes easier if you've done chapter 8. So chapter 8 is human. Basically, this is a new unit starting. So on chapter 7, the second unit ends, right? Reproduction was the first unit, and then second unit was genetics. Genetics end with evolution. And now we are starting in more applicative biology, which is biology in human wellness or something like that, human welfare. So we start with human health and diseases, where we study some important diseases and their concepts in human health. Okay, so we'll start with chapter 8. Okay, everyone, I think that will be the best chapter. Cool. So let's start with the basics. There are two things. First is health, and second is disease. So tell me what is health first. Health. What is a health? How will you define health?
State of complete physical, mental, and social wellness.
You forgot emotional. That's very important. Yes. So health is the overall well-being of a human. So we define in short as overall well-being of an organism. Okay, it can be human in case of humans. So we are talking about humans here because this is human health and disease. Okay. Now, what does this overall consists? It consists physical health, like your organs are functioning properly, the structure of your body is fine, and there's no problem with that. Then it's come mental and emotional. These two are slightly different because both have effects on behavior, but mental is with respect to the brain. Sometimes some problems, some neurological or biochemical issues with the brain can lead to some mental disorders or conditions where emotions or feelings become very overwhelming and you cannot control it. And then, as Nabil said, what was the third one? Nabil, you said social. Now, what is social well-being? Social well-being. What is social well-being? These are associated with diseases. What is social well-being? It's majorly associated with behavior, how we live in a society, how we live in groups as animals. Okay. So that's social well-being. So overall, it's known as the health. Is it clear, everyone?
Yes, sir.
Yeah. Now the second word is disease. Now, if you just look at the word, it's called dis-ease. What does it mean? You're not at ease, right? That's why it's diseased state. Disturb, disease. You understand? You're not at ease. So disease condition is basically where the physical or mental health is affected due to some factors. Okay, so it is the perturbed state. Perturbed state of physiology. Okay, or psychology. Both. Encompassing physical and mental health. Now also tell me, what are the factors that can affect health? So what all thing health can be affected by and can get into a diseased condition? Yes, people, tell me. What are the things that can affect health? Simple question. We have not even,
Pathogens.
sorry,
Environment.
Pathogens.
okay, come again. I could not hear you.
Pathogens, sir. Pathogen, parasites.
Okay, yes,
Environment,
pathogens and
climate.
parasites. Yes, so let's try to bin it. Let's try to put it in groups. You're working fine. Am I audible? Am I visible?
Yes, sir.
Okay. So someone said, let's go to Nabil first. Nabil said pathogens. So what do pathogens do basically in the body? How do they affect the health? They come and they create what? They do what? They infect, right? So can I club all of them as infections?
Yes, sir.
So infections due to pathogens, right? I'm going to say. So let's say infections are one way by which health can be affected. Yes, your answer, Saifullah, what were you saying?
Climate.
Climate. Mm, how? What about climate? Basically, you're talking about the environment, right?
Yes, sir.
Okay. So environment, but we are talking about abiotic component, right? Climate means not pathogens. Yes, because if you're saying microorganisms, then it comes here. So environment, for example, tell me what all things? Food, water. Yes. Can I also say with environment, can I also include lifestyle? Do you think that health can also get affected by lifestyle?
Yes, sir.
How?
Like if you are living in a very poor lifestyle, then there'll be a more
Yeah.
number of chances you'll be infected.
Yes, because rest, you might not get proper rest, or exercise, you might also not get proper exercise, which can cause obesity or other kind of metabolic disorders, right? Or sleep, lack of sleep also causes problems, right? Nutrition. So all these things, environment and lifestyle. Second region, infections, environment and lifestyle. What else? Yes, Mariam used the word genetic disorders. Very good, Mariam. Genetic disorders, right? We have just studied it the previous unit, genetics. And can you tell me what type of genetic disorders you're talking about? Mariam? Basically, it runs in the family. If it is genetic, then it's passed on from parents to the offspring, or it happened during the development of the embryo due to some mutations, right?
Yes, sir.
So write down. Yes. Defects. In this I can write defects. That is inherited from parents. Yes, defective genes basically. So defects inherited from parents or caused due to mutations or gene defects basically. So all these factors can affect health, correct? Okay, so let's talk about the first one now infections. Do you also know that all these factors that can affect our health can put us in disease condition? Okay, can put a human in a diseased state. Yes or no?
Yes, sir.
But this diseased state, it is of two types. You know that some diseases can be passed on, which means those diseases are infectious diseases. Have you heard of this term before?
Yes, sir.
Whereas other diseases are non-infectious, which means they cannot be passed. Like they cannot be, like if someone is a disease person and I'm in the vicinity, I cannot get that disease. That's not infectious. Can you give me some examples of infectious and non-infectious diseases?
Sir, COVID and mental health, sir.
COVID. COVID is, yes, it's an infectious disease. And mental health. Mental health is not a disease. Name the disease. COVID is an infection. So COVID is also not a disease actually, per se. Because in COVID, multiple things happen. We still are not able to figure out what actually it does. It just attacks the lungs, and the disease that happens, the problem that arises is that your SpO2 level goes very down and lungs get infected. Just like it gets infected in pneumonia, fluid gets filled and you cannot breathe and get heavy fever due to infection and all. But if we have to talk about a proper disease, let's say, which is infectious,
So then we
is,
can say
can we
AIDS
say
and schizophrenia.
yes, can we say STDs? Yes, AIDS, right? Sexually transmitted diseases are infectious, yes or no?
Yes,
They can
sir.
be transferred from one person to another. But if we talk about non-infectious,
Schizophrenia.
schizophrenia, yes, or can I say cancer? Let's stick to physical if we are sticking to physical, right?
Yes, sir.
So can we say cancer as a disease? Schizophrenia is a more than a disease, it's a disorder, neurological disorder. Yeah.
Sir, but in the
Diabetes
ending of
is
the
also
chapter, it
non-infectious.
says that there are some ways in which we can find out whether some people are more susceptible to cancer. So it is kind of
That's
like getting
susceptibility.
limited,
We
right?
all, yeah, but that's not infectious. That's susceptibility. If I am more susceptible to any disease, I may not give that susceptibility to my partner or my friend or anyone else.
So, but they
But
said that like if they look at the genes using molecular inheritance, they can find out whether in the family chain, based on his family chain, whether he is susceptible to some types
yes, so
specific type of
some,
cancer.
yes, some cancers run in families. That is what you mean to say. The susceptibility run through genes in the families. But again, it's non-infectious. It's coming through genetics. And if something is coming through genetics, it's not spreading through infection. It's spreading through genes. You know, that can even happen if the parents do not have that disease. For example, in case of, remember we were studying thalassemia and hemophilia, color blindness as genetic diseases.
Yes, sir.
So though parents were not affected, but still the kid can have those disorders and diseases, right?
Sickle cell anemia,
That's
right?
sickle cell anemia is also. So sickle cell anemia is also one problem that even if parents don't have, the kid can have. The parents are heterozygotes and the kid gets homozygous, it gets the disease. But still it's not infectious. It cannot spread from the kid to the nearby kid. Okay.
Yes, sir.
Yeah. Yes, someone said diabetes also. Who said diabetes?
Sir, I told.
Come again. Who's it? Madhya, Abhya? Madhya.
Yes, sir, I said
Okay.
diabetes.
Okay. Yes, and you're right. Non-infectious is diabetes. Okay. And another infectious, then you have to give me example of another infectious one.
So typhoid.
Typhoid. Okay. And what happens in typhoid basically?
Get fever, diarrhea.
And why is it infectious?
Can spread through in infected water.
Yes, it can even spread through infected food, right? So it needs to have that saliva containing the pathogen. Okay. Yes,
Sir,
typhoid.
even chicken pox is infectious, right?
Yes, it is. So all these diseases caused by, you understand it now, if they are caused by pathogens which can spread from one body to another, the disease become infectious. So do you see a trend? These diseases are either caused by viruses, bacteria, or some pathogen that can transfer from one body to another. But on the other hand, these diseases are not caused by pathogens. They are caused by genetic aberrations, cancer or mutations or something like that, right? So in diabetes, the gene for insulin is not becomes defective. Yes, it's not functional. It's not functioning properly. So that leads to diabetes. Make sense? And in
Yes, sir.
cancer, yeah, in cancer, again, it's either a mutation or because of misuse of carcinogenic substances like tobacco and alcohol. So it can have, one can have cancers in different tissues in different parts of the body. Okay, very well. So infectious and non-infectious, write down about infectious. Infectious diseases are capable of spreading are capable of spreading from one person to another. From one person to another. Infectious diseases are capable of spreading from one person to another through food, air, water contamination. Through food, air, water contamination, or exchange of body fluids. Or exchange of body fluids. Or exchange of body fluids. And continue. These infectious diseases are mostly caused by an infectious pathogen. An infectious pathogen. Am I audible to all of you? Clearly? Nabil, Madhya.
Yes, sir.
Sir, you are audible.
Yeah.
Yes, sir.
By an infectious pathogen. Right on. On the other hand, non-infectious diseases cannot spread from one person to another through exchange of body fluids or food, water, or air. Or food, water, or air. Now let's talk about some common diseases which are caused by pathogens and the stuff. So what are basically pathogens? Simple things. Foundation. What are pathogens?
These are microorganisms.
Okay. And they do what?
They infect a person.
And what happens during an infection? What do we call an what do we call an infection?
Disease.
No, but what happens in infection? One bacteria enters in your multicellular body, goes haywire. It entered through your nose and it created, you said it's microorganism. So how can one single tiny microorganism create so much of problem in the body?
They replicate, sir.
Multiplies.
They multiply.
Exactly. Yes. So something is a pathogen which is capable of multiplying in our body. You know, that's where we have to, it's very important to differentiate between pathogens and non-pathogens microbes. Every time when we eat, drink, we breathe, there are many bacterial spores, fungal spores, viruses entering in our body through surface contact, through food, through handshakes. We are actually exchanging lots and lots of microbiota. Okay, microbial life is being exchanged. Do you understand, everyone? And it is exchanged to such an extent that in evolution, many microbes have started making a relationship with us. They are in a symbiotic relationship. Do you know that all microbes are not bad that live and multiply in our body?
It's a lactobacillus.
Lactobacillus. Lactobacillus, does it live in our body?
Sir, I think it is, sir, in the intestine. I think.
Why do you think? What
I
does
read,
lactobacillus? It is
and
present in
Nikolai.
we eat it. It's a good bacteria. We eat it through our food, like curd and milk. It's there. But do we need it to have a symbiotic relationship with us?
No, sir.
No, right? So it is present. But some bacteria, are actually present
Equalize, sir.
as a E. coli. Actually, it stays, but we don't know, like E. coli has a good symbiotic relationship with us, because it also can cause sometimes infections. But other bacteria, good bacteria which we call, they make some essential products for us which our body can use. For example, vitamin B12, it's coming from our gut microbes. Okay, we have very limited sources of vitamin B12 in our body, so that is important. A lot of enzymes and other things are also digest certain toxins. That's why when someone has a diarrhea episode or loose motions, what do the doctors say?
Eat curd.
Eat curd. Why?
Lactobacillus.
Yeah, to replenish some good bacteria, right? Or probiotics. You have to drink probiotics. Anyways, if you don't eat or drink any of these two, over time, it's not like your diarrhea just cleaned every single bacteria from your gut, but it needs time for them to replicate and again repopulate in the concentration that is needed by us. So that's the good bacteria also live. But we call something as a pathogen which has the ability, write down, a pathogen is a disease causing organism or microorganism. A pathogen is a disease causing microorganism which that replicates in or on an organism and cause harm to it. Okay. Okay. Now, have you also heard of this one word called parasites?
Yes, sir.
What is a parasite?
Organism that lives and depends upon the body systems of the organism and also replicates in it.
Yeah. So in a way, if I ask you, are parasites, can I say parasites as pathogens?
Yes, yes, sir.
All?
Not exactly also. Some are not micro.
Like which kind of parasites I can call pathogens and which parasites I cannot call pathogens?
So the ones that are like protozoans are microorganisms like the malaria causing pathogens,
Yeah.
and fungi like ringworms, whereas macroorganisms are like roundworms and Ascaris Wuchereria.
Yeah, right. And let me also ask you a question, because you have brought up this point. Is malaria contagious? Can it spread from one person to another?
Yes, sir. Yes, sir.
No, it's not contagious. We can take care of a malarial person. We just don't have to get bitten by the person, like by the same mosquito that brings the blood, right?
Yes, sir.
So it's not like normally it's not contagious. It can be contained, right? So you can be near a person with malaria. On the other hand, flu is contagious, right?
Yes, sir.
Typhoid and flu, these are contagious. Because typhoid is caused by bacterium, and flu, viral flus I'm talking about, they are mostly contagious, or also your COVID is contagious. So write down pathogens. Pathogens can enter the body through various means. Pathogens can enter the human body through various means. What do? The next thing? They multiply and increase in number. Right? This is where they get the capability of causing infections. Otherwise, they cannot multiply and increase in number, no problem. And then they start producing toxins. They produce something, right, which is toxic to the human body. And these toxins interfere with normal physiological processes or activities. That's where the disease condition starts. And this leads to, these are just point one after the other. And when they start producing toxins, this lead to results in morphological or physiological or we can call functional, morphological and functional damages or issues. Is it clear, everyone?
Yes, sir.
Now, so in order to do all of this, in order to enter, replicate, produce toxin, result in some disease or damage, a pathogen also first has to adapt, right?
So what is it? Functional damages.
Damage. Some organ, something must start getting damaged, right? Function
Yes, sir.
starts, yeah. But in order to do all of this, a pathogen has to survive in our body, right? And to survive, because different parts of our body, what are the most probable? It cannot enter from anywhere. Like, what are the possible ways through which a pathogen can enter a human body? First, most obvious is through nose, the openings, nose, eyes, ears, mouth. Yeah. Or if there is a cut in the if there's a cut and pathogen, sorry, cut in the skin. Yes, Nabil, what were you saying?
Yes, sir. Cut in the skin.
Yes, and then pathogen can enter from there. So if there's a cut in the skin, we always have to sterilize the cut and not keep it open, right? So that's the point. They do not allow pathogens to grow around or get into the body through that cut. But when it gets into the body, what a pathogen has to do? It has to adapt to the environment within the body, right? Within the host.
Yes, sir.
And inside our body, in different organs, there are different environments. For example, a pathogen is causing severe diarrhea. Then where does the pathogen entering from?
In the gut.
Yes. Can a pathogen entering from nose cause something in the stomach or intestines?
No, sir. It has to enter through food or water.
Yes, it has to come through food or water and from the mouth. So in the stomach, we all know that we have very strong acid in the stomach. HCl, yes or no?
Yes, sir.
Yes, sir.
We also have digestive enzymes in our stomach and intestines. So if a pathogen is able to replicate, it comes from a contaminated food, it's able to cause infection in our stomach, which is known as food poisoning, stomach infection, or intestinal infection. What kind of features do that pathogen must be having? Add down the question. If a pathogen is capable, are you writing everyone the question?
Yes, sir.
If a pathogen is capable of causing infection in the stomach and intestines, if the pathogen is capable of having an or possible of causing an infection in the stomachs and intestines, then what are some characteristics of the pathogen which we can deduce? You tell me. Which we can deduce or infer or report, anything. Okay, let's start. Jasola.
Um, a pathogen should be immune to acids that is secreted by a body.
Exactly. The pathogen should somehow be able to survive in acids and enzymes, right? So write down. The pathogen that enters the stomach, the pathogen that enters the stomach should know a way to survive in acids and enzymes. Should know a way to survive in acids and enzymes. Okay. And also, pathogen should be able to tolerate low pH. Yes or no?
Sir, can you say again?
The pathogen should be able to tolerate low pH, because the pH in our stomach is very, very low. Yes or no?
Yes, sir.
Yes, sir.
Okay. Now let's come to some basic common pathogens and the disease that they cause. I think this is the life cycle of Plasmodium to cause malaria is there is much detail, and then we have to move into this chapter. Okay, let's talk about first. Let's start with what Nabil said. Let's start with typhoid. Nabil, can you tell what causes typhoid?
Infected food and water, sir.
What causes? What is there in the infected food and water? What is the pathogen? It's Salmonella typhi. This is the pathogen. Salmonella typhi. What is it? It is a pathogenic bacterium. and it causes typhoid for its name. Okay, typhoid fever. And write down certain things about these pathogens. So they enter the small intestine, as Nabil said, through food and water. Okay. Enter the small intestine through contaminated food and water. But some pathogens, when they enter the body, they attack the first organ where they enter. Okay. For example, let's say COVID. You know, it enters the body, it's there in the body, but it attacks the lungs majorly, right? Lungs is the major target of COVID. Typhoid, it also enters into the small intestine, but then it spreads to other organs through blood. Okay.
So,
Is it clear, everyone? So that is a property where they enter and then they start spreading to other parts of the body.
so like AIDS, so like it enters through the blood, attacks the liver cells and the immune system.
What enters through the blood? Sorry.
AIDS.
AIDS can
HIV.
also enter through body fluids like semen,
Yes, sir.
right? And then it enters into the blood to go to other parts, other organs. It basically lowers the immune system of the body, and immune system is through the mostly through the blood and the immune cells. Okay. Is it clear, everyone? So write down.
Yes, sir.
Yeah.
Okay, they enter the small intestine. Next, they migrate to other organs. So these bacteria migrate to other organs through blood. Okay. And what happens? What are the symptoms of this infection? Of Salmonella typhi infections? Can you talk about some symptoms? What happens in typhoid? First is sustained high fever, right? Fever is very high. It's around 39-40 degrees Celsius. Okay. Then weakness, of course, whenever there is fever as a symptom, weakness will follow. You'll never feel good in fever. So that's just easy thing to remember. But because the infection started from intestine, you will also have stomach ache. You also get fever in cold and flu, right? But you don't have stomach ache then. You understand, everyone?
Yes, sir.
Yes, sir.
And then you have constipation because there is infection going on in the intestines and you would not like to eat anything. So it's loss of appetite, etc. Okay. And in severe cases, which I'm writing here in red, very severe cases, if the intestine is very, very heavily infected, so intestines starts to break and we call it intestinal perforation. Okay, it's a severe symptom. Typhoid can be treated before it reaches the severe symptom, but if it's left untreated, or if the proper treatment is not done, then it can lead to that. So for every infection, what we have to study is what causes it. We have to understand the pathogen, what are the symptoms, and then we have to go to the test, confirmatory test. How will you know that there indeed is a typhoid infection in the body?
Fecal culture.
Sorry?
Fecal culture. Like you'll examine the feces of the person.
Okay. Okay. So you will take any, yeah, if it's a intestinal infection, very good idea. You can take the fecal culture and then look for the test in it. Okay. So the test which is done for typhoid is very famously known as Vidal test. Any idea about Vidal test? Anyone? Just remember this test. It is asked in entrances. So Vidal test for typhoid. What happens? Any idea what happens in Vidal test?
Sir, what is Manto test?
What is
Manto test.
Manto?
Yes, sir.
Manto will come to like you're answering it in context of typhoid, or you are just asking what it is. Mantox means, okay, so Mantox test, it's, if I, let me first confirm from you, it means this one, MNTOUX, right? This one.
Yes, sir.
Yeah, it's a basically skin test, okay, where there is a injection which we have to put in intradermal. The word intra and dermal makes sense to you?
Yes, sir. Inside the skin.
Yes, and which contains basically a very small amount of, like very small millimeters, 1/10 of a millimeter amount of tuberculin. Okay, and if there is a response against that chemical, if there is a bump in the skin, okay, where the fluid was injected, then it means that you may have tuberculosis infection in your body. Then you can go to, after you have this skin test positive, then you can go to doctor and get your TB test done through X-ray and other ways. So it is basically a tuberculosis test, skin test. Okay.
Yes, sir.
Because you have asked, so everyone other should also write. So Mantox test. Where did you read it?
Sir, they tested my mother, sir, for skin infections.
Okay. It's a skin test. I hope she is fine, right?
Yes, sir.
It came negative.
Yes, sir.
So it's skin test for TB, tuberculosis infection. So simple test. You inject something, small amount of tuberculin in the skin, and if it reacts, it shows a bump on the skin or red rashes, redness there. You have the test. You have the confirmatory test. But yes, let's first come to Vidal. So you know what is a Vidal test? So in this, write down Vidal test is a type of agglutination test which detects the presence of specific typhoid markers. That you have to know. Okay.
Yes, sir.
So basically, what you do is whenever there is, so for example, you said one way to confirm the presence of a pathogen, okay, right, Nabil, is culturing.
Yes, sir.
So if you do not have a facility to culturing you cannot do anywhere, right? To do culturing, you need a cell culture facility. You need to culturing facility which is free from any kind of contamination. Not even a single bacteria should be there. So culturing sometimes is time taking. The result comes in 3 to 4 days. It's also expensive, and everywhere it's not possible. So if you cannot confirm the presence of a pathogen or a bacteria by culturing it and increasing its number, what can you do? You can use this test, agglutination, which is an antibody based test. So the patient, you take patient's blood, okay? So write down Vidal test is a reliable diagnostic test for typhoid. Vidal test is a reliable diagnostic test for typhoid, which do not includes culturing the pathogen. Okay. So what you do in this is you take the blood, so write down, you take the patient's blood and
So
then
this is
from the blood,
method of knowing the list.
sorry,
So this
is
is the
the
method for Windle test.
yeah, it's how Vidal test is done. You do not culture it, so basically you just take blood, so write down the patient's blood and slash write down serum. Basically we need serum. The patient's blood or serum is tested for antibodies against the typhoid antigens, typhoid causing antigens. So the pathogens which are present, whatever they will secrete or their own things are known as antigens, right? So you just test for this. The specific antibodies which we are testing for is O and H type, but that's not important from your syllabus perspective. So basically what happens is when this bacteria enters the body, by the end of the first week, it rises very sharp, like it grows very sharp. Okay. And then it causes fever after some time. So there are two types of agglutinins, which are called O and H. So O can be detected early and H can be detected later on in when the infection has spread. So these tell you, if you can figure out O, and if you are not finding H, it still tells you that the typhoid is in its early stage. Then you can take a different route of treatment. So basically this is what. So Salmonella antigen, the pathogen's antigen, can be used to detect the disease. Okay. Is it clear?
Yes, sir.
Yeah. So the main principle of the Vidal test is that if there is an antibody present in patient serum, it will react with the respective antigen in the reagent that we are putting, and there will be clumping on the card. So if you put a person's blood and you put that antigen, and if there is both binding, then the blood will clump. There will be small clots present. So it's a very fast and reliable method. You just have to look at those clumps and you will get to know whether the person has typhoid or not instantly, very fast.
Sir, is it like the ELISA test?
Is it like ELISA test? Yeah, but you understand that they here and there in both, that's also an antibody characterization test, but it's called enzyme linked immunosorbent assay, ELISA, ELISA. Okay, and there also you are using it's a solid phase type of test where you basically use an enzyme immunoassay to detect the presence of ligand. So it's the same kind of thing, but the procedure is somewhat a little different. Okay, so there also you are actually using antibodies to detect the presence of a ligand. Is it clear? Here you are using antibodies to look for antigens. Is it clear? Both are blood tests, so it's kind of like that. You can understand.
Depends
Both
on what
are,
we're searching for.
sorry.
It depends on what we are searching for.
Exactly.
Right.
Yes. Yeah. So ELISA test is like you can do it for HIV. You can also do it for typhoid. You can also do it for dengue. Basically, it's just Vidal. You can say is a type of ELISA. You understand?
Yes, sir.
Enzyme-linked immunosorbent assay, but not exactly. So, do you understand the confirmatory test for this?
Yes, sir.
And I think there is a very interesting thing. I think it's given in NCRT. I was reading the NCRT a few days ago. So there was one cook who spread typhoid to hundreds of people unknowingly. She didn't know that she's a typhoid carrier. And she used to cook food for many people. And she continued to spread typhoid for several years through the food that she's preparing, and she's not knowing it. And people are falling ill. So when people started falling ill in big numbers due to the food that they were eating, then they realized that something is wrong. And the wrong thing was what was wrong was not in the food. It was that she was a carrier, and she was because when a person is making food, there's your breathing, and you have you're in close contact with the raw materials of the food, so you can just transfer the pathogen. You understand?
Yes, sir.
So I don't know her name. I know her name had Mary.
Typhoid Mary, sir.
Yeah, they started calling her Typhoid Mary after they figured her out. I don't know if that's a good thing to do, because it's like labeling a person as a disease, but I don't think she did it on purpose. She didn't know about it, but her name was Mary something, and
Mary
they
Mallon,
started
sir.
Mary Melanin.
Mary Mallon.
Mary Mallon, okay, and they started calling her Typhoid Mary. Okay, so this was just out of that thing. Okay, so that's it about the pathogen Salmonella typhimurium and the Vidal test. Another one is, which is in your syllabus, is pneumonia. You know what causes pneumonia? So there's another pathogen called Streptococcus pneumoniae. So this is the next pathogen that we will begin with. Streptococcus pneumoniae. So it causes pneumonia. The name is again typhi, causes typhoid, pneumoniae for pneumonia. Okay, and another one is called another Haemophilus. Yes, Haemophilus influenzae. This is also responsible for causing pneumonia. Both of these two causes, both of these two are responsible for the disease called pneumonia. Okay. So, oh, we don't have time. Time is over. Okay. 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 So, okay. So we'll take this in the next class: Streptococcus pneumoniae and Haemophilus influenzae. So, what is Streptococcus pneumoniae? A bacteria or a virus? Quickly, let's just discuss this part.
Bacteria.
Bacteria.
Bacteria. And what is Haemophilus influenzae? So look at the sorry.
Blood loving influenza.
Yes, what is it? Bacteria or what? Influenzae. Haemophilus means it's another bacteria. Okay. The word hemo means blood and philus means loving. So you know when it's cultured on blood agar media as bacteria. But don't get confused by the name influenza. Mostly students get confused thinking that influenza is like flu and flu is caused mostly by viruses, right?
Yes, sir.
So just so that you don't get confused, I thought it's good to bring it up. So about these two bacteria, we'll study in the next class. We don't have time now. So I'll start from here. We'll do pneumonia and also we'll go to some rhinoviruses. So we are doing Streptococcus pneumonia, Haemophilus influenzae, Typhimurium. Then we'll go to some rhinoviruses which cause the common cold. Someone brought up the topic common cold, right? That we will do. Then we'll go to Protozoa plasmodium, which causes malaria. And also the life cycle of plasmodium comes in detail in your exams. And then we'll go to some other worms, Ascaris and other things which causes Ascariasis, elephantiasis, and all these things. Okay. So in the next class we'll do all this. Then we'll come to immunity part for this chapter. Okay. So please read before you come to the next class. Please read before the immunity part from your NCERT. Okay. At least these pathogens. Okay, everyone. Cool.
So next class, no test, or will we be doing this?
It's a test. So tomorrow it's a test. Be prepared for chapter 6, and then after that we'll be doing it in the next class. Okay, so tomorrow there'll be just the test. Okay. All the best for the test, people. And please, everyone has to attend the test. So these are the series of tests started to prepare